We're now out of interphase and into mitosis, and we'll talk about each of the steps of mitosis individually. If I was teaching this class even 3 years ago, we would have talked about prophase and metaphase, prophase, metaphase, anaphase, telophase. The new fad is to actually divide this continuous process into an additional step, and that step is called prometaphase. It used to be called late prophase, or sort of lumped in with the end of prophase or the beginning of metaphase, but now it gets its own separate category. What happens in prophase? In prophase, first phase of mitosis, nuclear membrane disintegrates. The centromere with its attached kinetochore gets ready to attach to the spindle fibers. The chromosomes condense. They were in S and G2, they were spread out, they were transcriptionally active. Now, just like if you celebrate Christmas or you've ever dealt with light strings in a play or something like that, you know that if you just put those Christmas lights into a bag, they turn into an entirely horrible, horrible mess. But if you wrap them around and neatly package them, you can then pick them up and move them, and you can unwrap them without having a big tangled mess. So this is basically what's going on here. You're condensing your chromosomes and packing them so you can move them around. The other thing that happens is we get these regions known as centrosomes, and these regions are centrosomes, and these are the regions where the spindle fibers are going to come from. And the spindle fibers are the microtubules that attach to the chromosomes and are going to move them. Now, in human, well, in all eukaryotes that I know about, the spindle fibers, in fact, attach to the kinetochore, which is in the middle, or it's not always in the middle, but it's where the centromere is. In this case, it's being shown in the middle. In animals, there's an important structure known as the centriole, and the centriole is sort of interesting. So it is a set of two groupings of microtubules, and they're relatively short, and they sit perpendicular to each other. And during prophase, the old centriole divides in half, and a new centriole is, and the second group of microtubules is formed. So now you have those migrating apart from each other, and these are important in organizing the spindle fibers. But, and if you take them out of an animal cell, the animal cell can't organize and it can't do mitosis. In most plants, most fungi, most algae, they don't have centrioles, yet they do mitosis perfectly fine. And it's still one of those things that nobody quite understands, why this seems absolutely critical in animals but isn't needed in other cells. What does a cell in prophase actually look like? It is starting to become, the chromosomes are starting to become condensed, and individual chromosomes are starting to become visible, or at least regions of individual chromosomes. In this case, purple are the chromosomes and pink is the cell, or the cytoplasm of the cell. In prometaphase, this is where the kinetochore and the spindle fibers are fully engaged in interacting. The chromosomes are now starting to move around, whereas in prophase, they had become condensed to the point that they were short. They were pretty much spread randomly throughout the entire cell, and certainly the homologous chromosomes were not together or anything like that. In prometaphase, this is the stage where the cells are actually, not the cells, the chromosomes are actually pulled around heading for the middle of the cell. This cell here is probably in prophase. It's just starting to be condensed, for the chromosomes to become condensed, and these over here are in anaphase. Note they're still staining. The chromosomes are still extant. They still exist, and there are regions with more or less staining based on how condensed the chromatin is, but these are interphase cell, there's our prophase cell, there's our prometaphase cell. These, by the way, are onion cells, and if we were in lab, we could actually pull out and look at onion cells that are pre-stained. If you wanted to spend more time, you can actually stain your onion cells. It's not that hard to do.